Okay, so just to remind you, to connect you to last time, I was talking mostly about some biophysics, some biophysical implications of the fact. I hope I was convincing. I hope I convinced you that one should, or at least for certain purposes, one should consider the neuron as a distributed electrical unit rather than as an isopotential electrical unit. From the biophysical point of view, I gave you two examples whereby the assumption that it's a point neuron, if it would have been a point neuron, the electrical signals you would record from the cell body would look differently than if it would have been a distributed electrical system. And this implies that when you analyze certain data, recordings from the cell body, you should have the correct model in mind if you want to get a good estimation of some parameters like the time constant of the cell and other aspects like this. So at least I hope that this was convincing experimentally, and you know, we know today even to estimate how long a cell from the cell body out, if you would have measured the electrotonic distance from the cell body out to the tip of the dendrites, what would be the electrotonic distance, the cable length of this piece of dendrite. So it's about 1 lambda long, one space constant long from cell body to the tip of, let's say, the apical tree, depending of course on the condition. This is an in vitro condition. In the in vivo condition, yes, when you have a lot of synapses bombarding the cells, meaning that the dendrites are more shunted because of the activity of the synapses, then the effective RM, the effective membrane property, is less resistant. The membrane is more shunted. This means that when the cell is embedded in an active network, the electrotonic distance, the electrotonic length of a dendrite is much larger than in vitro. So the length could become 2, 3, 4 lambda long in the presence of many synapses bombarding the cell, which means that everything looks like, maybe you should consider the cell, you should see the movie that we did once with a student of mine, Albert. It's called the dancing neuron. Dancing neuron is a neuron that dances, not really dances, but when it receives a bombardment of inputs, it does like this, electrotonically, not morphologically, yes? Electrotonically, it does like this. So without activity, this is the length, electrotonic length, cable. And with a lot of activity, this is the length, and with a medium activity, this is the length. So it's a dynamical length unit. So the point is that in vitro, the minimum without any activity, cells are about 0.5, 0.2, or up to 1 lambda long, and depending on the activity, it may become longer and shorter, electrotonically. Also, of course, the time constant is dependent on activity, because time constant is Rm multiplied by Cm, and Rm, the membrane resistivity, which dictates the cable length, also play a role in the time constant. So as the network becomes more active, the effective time constant of the cell becomes shorter. So let's say in vitro you have a time constant of 20 milliseconds. In vivo you may get a time constant of 2 milliseconds. Okay, so it's a dynamical system, yes? It's not static, but I'm talking only, what I showed you was until now a very passive, static point of view about the neuron.
I'm not sure I understood, maybe I missed something
Okay.
about the longer electrotonically.
Yes. Okay.
You mean really longer, or
not
the electrotonic part?
Electrotonic, not of course the cell is not doing like this anatomically.
Yes.
No, I meant that if you have a cylinder, a cable, dendrite, that is 1 lambda long, let's say, in one condition, because Rm is something and Ri is something else, and so lambda is square root and proportional to the square root of Rm divided by Ri, yes? Divided by Ri. There is a diameter here. So I'm saying that when there is an activity on top of this neuron, so when synapses are walking on top of this dendrite, you know there are 10,000 synapses, or I don't know, many thousands of synapses, and And when they bombard the cell in vivo, the effective RM, this number becomes shorter, smaller, Yes, the membrane is more shunted. Because the membrane circuit, which is this one is becoming shorter. It increases the conductance, so the conductance increases with activity of the membrane, which means that the membrane resistivity is reduced, which means that lambda becomes shorter, which means that the electrotonic length becomes longer. So this is a physical length. Yes, let's say this is L, I don't know, 1 millimeter, and the electrotonic length L is L divided by lambda. So when I'm saying that this is 1 lambda, I mean that the electrotonic, the real length in microns divided by lambda is, let's say, 1. Let's say it's about 1 in the passive condition. But because lambda becomes shorter, when there is activity, lambda becomes shorter because Rm is reduced. The effective electrotonic length, L, becomes longer.
What's the meaning of the electrotonic?
The electrotonic means, for example, how much a synapse will attenuate from here to here. So that's what I showed you last time, yes? So if I have a long cable, the synapse will be seen like this at the cell body, and if it's a short cable, it will be attenuating less. So as more activity comes in, each synapse becomes a way, escape from the cell body, so to speak. It moves electrotonically away, and that means that the synapse is less effective. Sorry?
It means that there is more decay.
More decay. Effectively, because L becomes longer, so the cable length was 1 lambda before, and the same cable, the same morphology, because of the activity becomes, I don't know what, 3 lambda long. The same physical synapse here will say less to the cell body just because it became. But this is activity dependent, yes, depending on the activity. And so the time constant behaves also the same. L becomes longer, and tau m becomes, yes, the time constant of the membrane becomes smaller with activity.
That's basically what we mean.
It's a more coincidence detection than integration when you have a lot of synapses.
That's what we mean by shunting inhibition.
Yeah, inhibition, not necessarily inhibition, also excitation. Any synapse is shunting. The synapse is shunting, because it opens channels, independent whether it's inhibition or excitation. So as long as there are synapses active, they shunt the membrane, independent of the, I'm not talking about the voltage now that they generate, because if it's an excitatory synapse, the battery takes you up, and if it's inhibitory, it takes you down. I'm talking only about the cable properties, which depends only on Rm, Ri, and Cm, yes, the passive cable properties. Effectively, the cable becomes longer and shorter as a function of synaptic activity. So this is something you have to remember, that when you take into account a cell, you can look, if you look, for example, at Chaim's work on the tempotron, very interesting work. And initially they assumed that the cell has a fixed time constant, independent of activity, because the synapses were considered to be current synapses. But we know that synapses are not current synapses, they are conductance synapses. Eventually they inject current, but they also change the conductance of the membrane. So it completely changes the behavior of the system, because the time constant now depends on the synaptic activity. So it has a significant effect sometimes in the way the cell responds to the dynamics of the cell or plasticity of the cell, because it depends on the time constant, and this is a function of activity. But until now I actually didn't say anything about nonlinearity. I just wanted to mention that there are sources of nonlinearity, and of course synapse. The synapse is a dramatic source of nonlinearity in the system.
How does the electrotonic change actually changes our generalization model of the neuron? For example, with the longer electronic length, is it still valid to use compartmented model, or it's a better approximation to look at the neuron as one or two cells one unit. Because here, for example, here you can see compound mental model that does every small handwriting and so on. Obviously, if we can you go back a few slides? Or oops. Or a few slides.
This is the last slide on cable. So I don't know which slide.
You had
This is the cable equation.
with the keys, the slide with the keys.
With the keys. Oh, gee, you really look at the serious business here. Keys. Okay, this is the keys.
Okay, so
I see, this is what you remember from my talk, right? The keys. Okay, that's also important.
Reference because I couldn't. Okay, so as you talked about from point neuron to a more compound model, how does the change in the electronic affect the validity?
That's exactly what I was trying to show. That as soon as you introduce synaptic activity, we'll talk about nonlinear activity. I mean voltage-dependent activity soon, but even with synaptic activity, this becomes even more electrotonic distributed system. So with the synaptic activity, this is the correct direction, yes? Because the cell becomes more and more non-isopotential with activity. Yes, so without activity, let's say this is the point, I don't know what, 1 lambda, you can say, okay, 1 lambda is not so far from isopotential. Maybe. It's not true, but let's say. But when this becomes 3 lambda long, then it's even more difficult to justify that I neglect electrotonic structure. So activity emphasizes, synaptic activity emphasizes the electrotonic structure because it makes the cell electrotonically longer.
But on the other hand, because there's really fast decay,
Fast decay, that's another thing. So we are talking about 2 things. We are talking about the electrotonic length, which is a space unit, yes? And we can also speak about the time constant of the cell, which becomes shorter, you're right. That means that the cell becomes more of a coincidence detector. You have to really time the inputs because the time constant becomes so short. If things are not timed correctly, then you don't sum. Yes, so that's another issue, but it doesn't have anything to do. So this is just a special. What I'm representing here is just a special representation of a cell. Is this space can be lumped into a point? So this was the original question. We didn't discuss time yet at all. That's another issue. The timing of synapses, the coincidence detection, the summation of synapses. This is not part of this drawing here. Yes. We didn't speak about the time of the keys. We just spoke about the details of how many details you want to take to describe this in space, the cell. So we didn't speak about time much. I was just mentioning here, I didn't want to go into it much, but I just wanted to embed on you that the cell is not really something that you should consider as a tree outside. Yes, because the tree outside is not so much affected by the fact that I don't know what, it's not an electrical creature, but this is an electrical creature, and this electrical creature depends on the electrical activity, and in most cases you get a lot of synapses active, and this means that it does like this. So our dancing neuron movie is really reflecting the fact that depending on activity, the neuron is dancing, and depending on the music, if it will be a Beatles or something else, depending on the music that bombards the cell will start to dance, electrotonically dance, it's an electrotonic dance. According to this. Okay, let me continue. Is it clear, or is there some fundamental issue that we'll talk about time later, but the length is not fixed, electrotonic length. Of course, the cell can also grow morphologically, spines and all that, but this is not what I'm talking about now. The other word, if you already talked about electrotonic length, maybe I should have mentioned, I just jumped last time because of the limitation of time. So you see that you also should, of course, you can also define space constant as a function of frequency. lambda as a function of omega. So in this case, the typical classical lambda space constant is for the steady state case. So everything is steady state, yes? That's the graphs I showed you. The steady state input, I is steady state, and you see the voltage, the steady state voltage attenuation in the steady state case, so there is no time. But you can also define space constant as a function of frequency. So for example, if this is the electrotonic length of a cell in vivo in steady state, the same cell would electrotonically look different depending on the input frequency. For example, if I will inject here 100 hertz sine wave into the cell body, the attenuation, let's say in units of 1 over E, will become, of course, because of the capacitance, it will become more and more attenuated as the input frequency increases. So effectively the tree, the electrotonic length of the tree, and that's the dancing neuron, yes? The dancing neuron in this case is dancing in terms of electrotonic length depending on the frequencies that it sees if you want to measure the cell in units of attenuation. So if you ask yourself, I give 100 hertz here, how much of it will be seen at this distance? Because of course when the frequency will be larger, when the increase in frequency, it will be attenuating more because of the capacitance. So what I said before was a steady state description of the cell. Of course, the cells see frequencies that are not steady state, so they see many frequencies, and this means that in terms of attenuation, what we call the attenuogram or attenuation gram, you can see the cell looks like this. Not because there is a change in conductance, just because the input is fast, and because of the filtering effect of the cable, there will be more attenuation of the input as the input becomes more modulated in higher frequency. Anyway, so just not to think about the cell as something that is static, yes? Fixed.
The range of frequencies that can
That the cell might
cause
receive.
steady state.
So the term steady state here is related to the time constant. So let's say that the time constant is 20 milliseconds in some condition. So if you have an input, let's say NMDA input, that the time constant of the NMDA input is relative to the time constant of the membrane much longer, let's say 4 times longer, I would say the NMDA input is close to steady state. If it would have been an AMPA input, which is fast, compared to the time constant, I would not call it anymore a steady state input, and then I have to compute what is the relevant frequency of this AMPA. Maybe it's 100 hertz or something like that. Okay, so the attenuation depends on the frequency of the input, the degree of attenuation, which is clear, of course, because you have also time here, not only space. Okay, but again, I can't go over. This is cable equation, so all that I said is actually not very new to you, in principle at least. So let me go on and just mention that as soon as you want to describe more refined local geometries, for example, the dendritic spine, this is a reconstruction of a piece of a cortical pyramidal cell dendrite. And you can see, of course, the amazing structure of spines, which are little cables. Yes, short cables emerge. I mentioned them the other day. I told them that this is a very thin cable. Now, this is from electron microscopy, reconstruction under electron microscopy. But of course, you can see the spines in EM, in light microscope. You cannot what? Not like that. This is really not only like that, but if you really want to see the synapse sitting here, you have to section it with EM and reconstruct here. But if you really want to see, okay, so now I want to show you something that I didn't mean to show you, but I'm very enthusiastic about this beautiful picture. Let's see if I find it. So I was just visiting the Human Brain Project, the Blue Brain Project, which we will not have time to discuss. And there was this anatomist, Javier de Felipe, that I mentioned, that is absolutely in love with neurons, very much in love with neurons, which is good. Very good to be in love with neurons. And so he gave me a new slide of his, because he used a light microscope to reconstruct human brain cells. Human brain cells, in addition to giraffe and other brain cells. So he sent me these two. So this is Monica Hall. As I told you, this is a grandfather of every anatomist, but every Spanish anatomist. And you know that spines were discovered by Ramon y Cajal. So he didn't call them spines, protrusions he called them, but anyway, so Ramon y Cajal already saw in his microscope that there are these little things. This is with a very primitive microscope that I mentioned before. But today, with a much more sophisticated microscopes, you can section a dead brain of human, in this case. You can inject this fluorescent dye into single cell in the fixed tissue. It's a fixed tissue. You can impale with an electrode with a fluorescent dye, because he's interested in comparing the human spines to, let's say, the mouse spine. What makes us human? What makes us human in terms of morphology? It's a good question. So you can see this is really light microscope. This is not EM. This is light microscope of single individual cells injected in the slice of this human brain. So in his recent paper, he compared a brain of a man that died at 45 and a brain of a man that died at 85. N equal 2, 1 and 1, to compare their spines. So this is what you can see. This is now light microscope of a dead human tissue. You can see really individual spines. And you can even zoom more with light microscope. And you can see this is absolutely amazing figure in my mind. Really amazing. And he calls this, what did you ask?
Memory.
So yeah, so I don't want to discuss it now, of course. But yes, so he calls this, so this is a figure. Then there is an interpretation, but this is a figure from a brain. This is a pyramidal cell, human pyramidal neuron, and then he calls this memory spines, and this he calls learning spines. This is a debatable issue, but he claims, he's also doing a lot of EM. He claims that these spines are those spines that are growing and not yet making synapses, but they would make synapses under certain conditions, and we don't know, of course, the algorithm when this little philopodia, this little branch, is making or not making a connection. But when it does make a connection, it looks like this. This is true. So it becomes a real spine, so to speak, yes? So there is now a synapse, there are receptors here, and presynaptically, as we mentioned last time, there are the axons coming. So he calls these are the learning spines because they are trying to learn something and make connections.
He claims they keep doing that throughout.
Yeah, we know that they are keeping doing
doing
it.
that.
Yeah, there is beautiful work now that we can do it, in vivo, looking at spines in vivo during behavior. We can look at individual spine and see that the spine is doing like this, and then tap a synapse. So this is why I call it learning. But I just wanted to show you the amazing level of anatomy that you can do today on in this case dead tissue, but we can do the same for dead tissue. But
Is this the 45-year-old
sorry?
person or the 85-year-old?
Sorry?
Is this from the 45-year-old?
Oh, gee, this I don't remember. I think this is the younger man, yes. He showed that the older man has, of course, less spines, less connections, but it doesn't, these are one by one. I don't know if you would take Shimon Peres, our president. He certainly may have more, I'm sure he has more spines than Bibi Netanyahu, is younger. But that we didn't check yet. Anyway.
Weaker spine.
Weaker spine. Not very creative spines in one case. Okay. So the question is, how do you model, so to speak, this kind of protrusions of these branches? And of course, in this case, you also can use the cable equations because these are small cables, and you can really reconstruct the cell and solve the cable equations. But in this case, you will have many branches, and each one with its own property. But typically, what people do today, like myself and many others, in case that you want really to ask questions related to individual synapses working on this spine, then how do they communicate, let's say, with another synapse sitting on this spine, and what is the, voltage here when these four spines are active. If you want to know something about local activity in such a detailed branch system with synapses impinging on the head of the spine, then you typically go to the compartmental modeling approach. and you represent each of those compartments with, you know, the head of the spine would be an RC. Depending, of course, on the parameters, you would give real values to the capacitance of this, to the passive resistance here. There will be a synapse here, maybe NMDA synapse or alpha synapse, so it will be a time dependent or voltage and time dependent, depending whether NMDA or not. And so you can really inject the synapse, so to speak, here, and you can really see what would be this neck doing, this neck resistance doing to the dendrites and so on. So compartmental modeling is the basic tool today in single neuron modeling, is the basic tool today to explore issue of connectivity, of crosstalk, so to speak, crosstalk and local influence in a complicated dendritic structures like this. Because you know, and that's of course one aspect, is that there is a lot of local plasticity, for example, in a group of synapses that may work here, they may undergo plasticity locally, independent of the spike that may or not may happening in the cell body. There is a lot of interest in understanding what happens locally when a group of synapses locally are active continuously. What happens? Who is the teacher? It's not STDP anymore because there is no spike necessarily. So this is a lot of local interest today about what happens locally in spines and dendrites. Also because the technology enables us, as I said before, to in vivo look at local dendrites locally. We're doing imaging, and I mentioned that you can even image the directional selective of an input, and we shall hear it from you next time we meet about what can you do with imaging local input to individual spines using two-photon microscope and local plasticity as well. Anyway, so okay. So I promised you to mention nonlinear dendrites and just the history of it. So about 25 or maybe 30 years by now, there was this development of the DIC, differential interface contrast video microscopy. And this is, by the way, in America, totally not allowed to say DIC. Maybe now with Obama it will be okay, but it was not allowed because of all these connotations. But here I allow myself to say DIC video microscopy, and this DIC video microscopy is showing you the cells, and so people started, because of this DIC video microscopy, to be able to patch, because you can see the dendrite. Of course, you can zoom. I'll just give you one optical slice. You can, of course, you can see the dendrites of this. You can see this pyramidal cell, layer 5. You can see the dendrite, the apical dendrite going this way, even in this bad resolution. And so you can start to patch. And I told you that people started to patch dendrites about 30 years ago. And today we can really see what happens in the dendrites due to this.
Where is the video?
Sorry?
It's supposed to be a video?
Yes. It is a video, and it is an optical technique to enhance the contrast. And so now every lab has it, of course, but this was a big revelation. This was before the two-photon microscope. So the name of it is, I don't completely myself understand how they treat the system so that you can really see such these are slices, yes, these are slices, cortical slices this way. And so this gave the first time the capability for Bert Sackman, who I mentioned will be here next week, and with Greg Stewart, and this is a really dramatic Nature paper because it was the first time they succeeded to systematically and by visually directed patching dendrites and soma at the same time, same neuron. So they just wanted to prove that it's the same neuron, because you may know that it may not be the same neuron, because you may patch a dendrite of this one and the soma of this one, but how do you know that this is the same? Because you don't see all the way through, so to speak, yes? So there are tricks to do it. You can inject current here and see where there and there and there. In this case, they inject dyes here and dye here to see that they meet. Anyway, this is a very beautiful paper from '94, but this was not the purpose. The purpose was to see what happens or where, so to speak, where does the action potential is initiated, and does it go backward or not. So the term back-propagating action potential, which I'm sure you heard, the back-propagating action potential came from this paper. You know this paper, so I don't need to talk about this at all? You don't. So, okay, by the way, this is the axon. Again, this is not a good resolution, but this is the axon, and this is the beginning of the basal dendrites. So remember, there is the basal dendrite, and this is the stem, the main stem of the apical dendrite, and these are the oblique dendrites. and if you would go there, there would be this tuft. Okay, so they record here and they record there, and they did something really beautiful in this paper, and this is the following, just to tell you about a little history of Stuart and Satman. And so in this case, so they can inject the current here, or they can inject the current here. They can also record here. This is a little later. So they have 3 points of recording in the same cell, and you can see that, and that was interesting, that when you injected the current at the cell body, when you injected the current, when you stimulated only at the apical tree here, so you see the voltage grows here, so the voltage starts here, it attenuates to the cell body, yes, the cable equation, it attenuates to the cell body from here to here, so you see the recording at the cell body, but then you suddenly see the burst of the jump, so to speak, of the spike at the cell body, although you inject here. So it was attenuated, but still it fires first at the cell body. So this was a direct proof that although the synapses, the input is in the dendrite, the action potential started the soma, and actually when you recorded the axon, you can see that it actually started the axon. So it first starts here, then it attenuates here, then it attenuates there. Okay, so this is the sequence of the timing event. Starting at the axon, going back into the soma. Typically, until this point, we recorded only in the soma, because that's where we could record before. So we saw the action potential here, but we didn't really know where things start and where do they go. So they saw this, and this is the interesting thing, because this we actually knew before, that the axon is the hotspot. And so the hotspot is the one to ignite first. But then they saw this. This was interesting. Why? Because this is very large. This is really big. So this is a big signal related to the fact that you have an action potential here, and that's where the ingenious part came in this paper. They said, but how much of this is active propagation from the cell body backward, and how much of this is just what expected from passive propagation? Because you have a big signal here, a 100 millivolt signal here, it would attenuate backward because it's a cable, it attenuates anywhere. So maybe it will just a passive thing. So what they did, and that's the ingenious thing, they put TTX, and they knew that this is a sodium spike. So we know that this is depending on the sodium channel, open of voltage-dependent sodium channel. They also knew, I don't have time to talk about it, but these sodium channels that are very hot in the axon also appear in the dendrites, so the dendrites also have voltage-dependent sodium channels. We knew that, but how much of them, what is the density, and so on. So anyway, they pour TTX to the whole preparation, so there are no spikes anymore, no sodium spikes anymore. But still, they want to see what would have been the passive attenuation, so they voltage clamped the cell body with the shape of a spike. So the voltage clamped the cell body. They inject a voltage to the cell body, not current, voltage to the cell body by voltage clamping system, fast voltage clamping systems that imitate, replicate, copy. They copy the recorded action potential, exactly this action potential. They recorded this action potential before, and now they injected this action potential, so to speak, by replicating the shape of the action potential to the cell body. Now the cell is passive. So it cannot generate its own spikes. It's passive. And then they ask how much of this action potential that now I replicate in the cell body would be seen at this location. This is a very nice trick, yes, to overcome the fact that you cannot have spikes, but you can inject spikes, so to speak. So they called it a simulated spike. I mean, simulated not in the mathematics, but simulated in the external.
How you know if it's passive or active?
So they blocked active channels.
Blocking with chemicals.
With TTX. So TTX blocked at least sodium-dependent. Voltage-dependent sodium channels are now blocked all over. And so there are maybe other voltage-dependent channels, but that's another thing. So they block the tentative channels that may generate the action potential. There is no action potential in the cell body. There is no action potential anymore in the axon. So they generate this action potential, and they see, I forgot to bring you this slide, and they see that, that if it would have been completely passive, without this voltage-dependent sodium channel, you will see something very small like this, at this location. So the difference between this little small thing and what you really see is carried by active channels in the dendrites. This was a direct proof that there is a boosting, there is an amplification of the action potential that starts here back into the dendrites, and that's why they call it backward propagating action potential. It's not spreading or attenuating, it's propagating action potential, actively propagating action potential in this direction, but it's not fully propagated. So it's a little bit weak action potential, sick action potential, semi-action potential. So it's a new signal in the nervous system. We did not know about this signal before. We did not know that there is, we thought there is passive signals, synapses, action potential, and this is a new signal, so to speak, because it is in between passive, it's not passive, and it's not completely active, it's a semi-active, or I don't know how you want to call it, weakly active action potential. So this is a weakly active potential, but it's weakly, but still quite strong, because several hundred of microns, let's say here and here, you may see 50% of the original one, which is big. So it could be 50 millivolts. 50 millivolts is a very big voltage, yes? Synapses have a hard time to generate 50 millivolts. So this is an important thing. Of course, there is a big, again, I don't want to go into it because this is a very basic biophysics, and I want to go to the computational aspect. Just to tell you that this was, for me at least, I was very impressed by this paper, not only me. I almost wanted to give Sackman second Nobel Prize because of this, because he got already Nobel Prize in 1992. But this is really shocking paper, I thought. And the fact, and the interesting thing, and that's one thing, of course, you all know, that this backpropagating action potential is a very important signal for handshaking between the synapses that generated this action potential and the signal going back to the synapses, telling the synapses that they succeeded to generate an action potential. So this was a classical question. You have a synapse here. How does this synapse get a feedback somehow, a teacher somehow, that it happened here because it's remote? So the backpropagating action potential is a very important signal. We know plastically that if you block it, there are beautiful work on blocking the propagation backward locally. So you can spritz TTX here, only here. Yes? So to avoid the backpropagation, but not to avoid the forward propagation, but only to avoid the backpropagation. And you can see that synapses do not succeed to learn, so to speak. So they need this signal, at least these local synapses here. By the way, this action potential also goes to the other dendrites, yes? I'm only showing here. And then there was a question, but what about these distal synapses, because this action potential does not reach there. This is too long, it's away, so it attenuates, and then we have calcium signal here. So there are all these, the many features. I may get to it in my second talk, in my second part, because there are many local plasticity-inducing signals. And the backpropagation action potential is one of them, but not only of them. So that's one thing. The other question that was really immediately, for me, an interesting one, and so we wrote a paper to the PNAS to ask the question, but why doesn't it start here? How come, so you have an active channel here, but still it doesn't start locally, it starts elsewhere, and it propagates here, yes? But it doesn't propagate backwards, so it means that there is an active propagation, so why isn't there active propagation this direction, and there is only active propagation this direction? It's the same channels. How does the channel know if it comes from here or here? Yes? So you have to show mathematically or analytically, right. Semi-analytically is, of course, when you have a very big signal happening here, like 100 millivolts, this 100 millivolts is sufficiently strong to activate in this direction the channels. If I would have injected 100 millivolts here, it doesn't happen in reality here, but if I would have injected a very, very powerful signal here, then I would activate a spike locally as well. It sometimes happens also in biology, but typically not. So typically this is the case. It always starts here, backpropagate there, start at the axon, go to the soma, back to the dendrites, and so on. So that's the story of this type of nonlinearity, which is only one type of nonlinearity in the layer 5 pyramidal cell. By the way, this is not always the case. Not every dendrite type have this backpropagating action potential. For example, Purkinje cells in the cerebellum do not have it. There, the dendrites do not have this backpropagation action potential. They have calcium spikes in the dendrites, which is something else. And this is just to start to impress on you that dendrites are highly nonlinear elements. They are not passive, absolutely not passive. In some direction they be more passive, in other more functionally. Yes, functionally they are more active in this direction, less active in this direction for this particular case. But just to put to show you that this was a very direct example, and also in the same paper they also succeeded to what is called the excise patch, to take a little patch from the dendrites with the electrode, so to suck a little patch of membrane from here, and to directly measure the density of channels in this little patch. So really they could measure the density of sodium channels in the membrane using this little patch. So you excite the patch, and this patch is glued now to the electrode, and you can really record individual ion channels opening and closing in this little patch. And we know today that the density of sodium channels in the apical tree membrane is about 30 times less than the density of the channels in the cell body. So it's not as dense, and that's why the action potential is not. That's why the dendrite is not an axon. Because if the dendrite would have been completely an axon, this spike would flush. Yes, it flush the dendrite, will be more difficult to integrate. It will reset the dendrite each time it goes there. It will be more difficult to integrate. So anyway, this is a dendrite, this is an axon, also in terms of density of sodium channels. On the other hand, there are many channels here, ion channels, the voltage-dependent channels here, they do not exist. They do not exist in the axon. There is a sharing of labor or something. Okay? So this is the starting of the nonlinearity, and the other big thing, of course, is this another fantastic work from another student. So this is Larkum, Matthew Larkum, who is now in Berlin. So he succeeded to record even more distally in the layer 5 pyramidal cell. This is the red electrode, and you can see that they record in three locations. And this is a classical figure showing that if you activate with a low intensity of injection of current locally here, you will get a local, so to speak, simulated EPSP. You inject current, you get a voltage as expected. When you stimulate the cell body, so this is the black electrode. When you stimulate the cell body, you will get a spike as expected in the cell body. This spike attenuates backward, as I mentioned before. So you will see this back. You don't see it maybe very well. This is this magenta. This is this blue, white blue, and you will see the backpropagation here also. So in this case, the backpropagating spike succeeded to get up until here quite well. But you can see a little bit here, this hump. And this is a calcium hump, so to speak, because we know that if you time the two inputs, the distal one, like here, plus the somatic one, like here, together, with a certain time delay window, like here. So this is the somatic injection, this is the distal injection. Suddenly, and so you get the spike here, suddenly you get a huge red calcium spike here. Okay, and as a consequence of this huge red local hotspot calcium spike here, you get a burst of sodium spike at the cell body. So this is the first one that emerged from the direct injection, and this tac-tac, these extra two spikes, are the consequence of this large calcium spike in the dendrite. So there is this ping-pong effect, because the backpropagation action potential goes here, together with local injection, boost locally, you get a calcium spike, and this huge inward charge propagates passively, because the action potential here doesn't, the calcium action potential doesn't propagate fully here. There are no calcium channels here, only locally here. So, but there is a lot of inward current, which adds current because of the cable. There is attenuation, but still enough to get extra two spikes. So this is what I call the ping pong, because this goes there, ignite this, and this goes here, and ignite this. Yes, and it's a ping pong. ping pong between two hotspots. It's a ping pong between the hotspot here, a sodium hotspot, and this is a hotspot of calcium hotspot, and they interact between each other. And they boost the system depending on the time window of the two activations. So they took it very far. They said, this is a signal, this burst, which is very difficult to get otherwise, is a signal output. It's a readout signal. It's a readout signal. The two events happen together. There was an input to the basal tree. This is the input to the basal tree, let's call it. This is the input to the apical tree, distal apical tree. When they come together in a certain window, then there is a readout.
But it's opposite now.
Opposite?
Like it's supposed to be the other way around. The red is supposed to come before the spike, and then it's supposed to return. Like
What do you mean the red is supposed to come before?
like in
You mean
learning.
in life? You mean in
Yeah.
life? In life.
In learning.
Okay. You mean the top-down, this is a top-down input, yes? Because layer 1, you mean that layer 1 receives input from...
Yeah, you retrieve input, and if that input is sufficient enough to ignite an action potential, then you see a backpropagation. And then, like that.
No. So the claim is the following. Suppose there are enough synapses working here from, let's call it bottom up, let's say from the thalamus, I don't know where. There is a lot of input coming here. So this is this sodium spike, this is the first one. So there is an input coming from here. Let's say you see me, it comes from here. Then there is the memory that comes from there, yeah, so to speak, of me. So this coincidence of the input coming, let's say, from the environment, this is just a metaphor here, yes? Input coming here, igniting the local spike at the same soma. If at the same time there is also, or not the same, a little bit later, if a little bit later there is also an input coming here, then the output of this cell will send the message that these two things happen together, or not together, but with a certain time window. And then so this will be a signature of this coincidence of bottom-up, top-down, coincidence of certain. There are a lot of theoretical work that came from this, about this, what can a cell do because of these nonlinearities and the ping-pong effect, about saying that this and this happens to it. What kind of coincidence detection can you do with this nonlinearity? So again, I don't have time just to go into the details, but just to, as I said, I just wanted to give you an intuition about what can you do with a distributed system, and in this case I already talked about nonlinear distributed system because there is nonlinearity here, there is nonlinearity here, calcium nonlinearity and sodium nonlinearity, and these two may interact. For computational reasons maybe, I'll show you later, we'll go into it, that we can also ask about plasticity here. Because you have two teachers now, yes? You have one teacher that teaches these synapses, because these are the pupils. Those that generated it are not generated, but those that are involved in generation of synapses that work here, near the hotspot. And this sodium spike is the teacher of these synapses. So there are these two classes of synapses. There is the proximal class of synapses listen maybe mostly to this one, but they interact. So this interaction may be, if there was another class there with the teacher, I was teaching you, Chaim was teaching them, and I was talking with him, and you hear half of him and half of me. It's not clear what you will learn eventually. So that's the question here about two teachers in the same neuron. So this is another project that we almost just finished, but I cannot tell you everything, but I'm just highlighting. Okay, so before we take a rest a little bit, I just want to go to the cortex, just to mention two things with the cortex. And next year we'll give with Chaim a whole course about cortex. But so I'm giving you a 5-minute lecture about cortex, just because we want to go later on into some aspects and tell you what happens today about the cortex. This is, of course, part of my beloved Blue Brain project, which is very controversial. I cannot mention Blue Brain project near Chaim because he's going to kill me, or any physicist actually, but of course they are wrong. And so I want to show you this diversity of cell types in the cortex, very, very beautiful morphologies, and many types of neurons, especially interneurons, beautiful different morphologies. And today, because of the Blue Brain project, I myself have learned that there are about 56 types of morphologically different cell types in the cortex, of mammalian cortex. So of course this is a draft. There may be one or two more or so, but just to highlight this, and then we'll talk about this later. So this is layer 1, layer 2, layer 6. This is the cell types, and they have names, and I'm not going to go into each name here. But note that, for example, in layer 1, there are only inhibitory neurons. So this line distinguishes between excitatory neurons, mostly pyramidal cell, but not only, and all these are inhibitory neurons in terms of morphology. This is a morphological classification. Yes? They have different trees, different faces. Now, one second, and I'll answer just to tell you this cloud that you see, the cloud that you see is a cloud of many, many neurons of this type superimposed. So it's, of course, it's not the same morphology period. You cannot superimpose them. So this is the cloud of occupancy, the cloud of cells of this type, the cloudiness, yes? And so, for example, this is layer 2-3 pyramidal cell, layer 2-3 pyramidal cell. This is layer 4 spiny stellate cell, spiny pyramidal cell, pyramidal cell. These are 3 types of excitatory cells in this layer and so on. So today we know in terms of morphology that there are all these different morphology. Whether it's important or not, that's another issue. But if you do objective classification, if you take all this and throw it into a classifier, without us saying anything, it will classify it automatically into 56 types. Today, I'm not saying that we succeeded to reconstruct. This is, by the way, 15 years of work of one person, that she is really amazing, a woman from Japan who used to work with Henry Markham. She's now a professor in America, and she's an anatomist. And she really liked this classification of morphologies. She's not an electrophysiologist, she's an anatomist. And this is really 15 work of thousands of cells, thousands and thousands of cells reconstructed by hand, and then put together into our systematic classification system. And so I'll give you now the updated version of what we think today about the cortical column.
Do you have also a probabilistic map of such a map?
Yeah, so these are the probabilistic maps. Yeah?
No, I mean like how frequent one might
Yeah, of
find
course. Again, from our measurements, I'm saying that several thousand is nothing relative to what you should have done, but that's what we can do today. Yes, we do have probabilistic, and we have something like this today. Again, I just show you what we, you know that we applied for a 1 billion euro grant last 2 weeks ago. Not that we are going to get it, first because Europe is collapsing, but also because we have huge competitors on this 1 billion. But we are two out of 6 will get 1 billion if Europe will be able to give 1 billion. But so we are one of 6. But anyway, it doesn't matter if we get the billion or not get the billion. That's not the important thing. The important thing is that we learn something about the cortex. What?
Why not dividing between all the 6?
That's a good question. That's a very democratic question. Socialistic question. That's a socialistic question. Yes. There is a huge competition. I guess the one that will win for sure is the Nobel laureate, that the physicist, the Russian physicist who lives in London, in England, who invented or found graphene, the graphene world. So he's one of the competitors. Graphene. Doesn't have to do with brain, by the way. Not the whole 6 are brain. We are the only one on brain.
His name is Novoselov.
You are the Russian fellow. I don't even pronounce his name. But yes, I'm sure you're right. Yeah, okay, so he's good. He's good. He's very good. He's absolutely good. Yes, very good. So his project is to develop new nanotechnology, and he already proved that just by playing you can do this one monolayer, atomic scale wires and so on. Anyway, so I don't want to go into these flagships, but this is part of the flagship. I just wanted to tell you that I myself, and I hope you will next year, and Chaim and us together will try to learn more about what we know today about the cortex. And so I'm just showing you some anatomy that came from all this reconstruction and all this classification. And so we, for example, know something about the proportions of cell of inhibition versus excitation in each layer. So this is statistics of numbers, not of types, of numbers. So So you can see here, you see,
Numbers
this
in
is layer one, numbers, physical numbers per layer of each type.
region or
No, so okay. So I can't go into it now because you need a little break, but for example here you can see, but there are several colors. And each color codes for different inhibitory cell type. So I can tell you that this is the proportion of this inhibitory cell type, and this is the proportion in this layer 1. And if you look at layer 2, 3, for example, here, I can tell you the proportion between inhibition and excitation. So here it's only inhibition, yes? So the proportion between inhibition and excitation is 1, there is only inhibition. So I can tell you the proportion between inhibition and excitation in each layer. This is a broad classification between excitation and inhibition in terms of total number per layer. But I can also tell you within layer of each type, how many of this type in percentage is here or there and so on. So that's part of the project, is to really classify and build in silico digital model, so to speak, of the columns. So I just give you the latest, the version that was submitted. It's part of a 380 pages, 1 billion cost a lot of pages too. And anyway, so this we start to know more and more about the local structure. You see there is a lot of diversity, a lot of local classification types, which are not uniform, neither in numbers, and so on. And of course, we absolutely, completely do not understand why nature invested so much effort. So it seems, unless you tell me it's a bug, why did nature invested so much effort into all these kind of subtypes, chemically and so forth, but not only chemically, but of course electrically, because you realize what I said just before, and I'll finish with that, that each cell type, morphological cell type, also have an electrotonic signature, an electrical signature. Yes, not one by one, but let's say parvalbumin type, I don't know what cell, inhibitory cell, may be this kind of electrical type. It will respond to square pulse by doing that, or somebody else by doing that, and so on. So there are all these electrical types, which are not mapped one-to-one to the morphological types. So, yes? So, for example, you can find this non-accommodating action potential type in many different cell morphological types. Yes? So, but we know today how many morphological electrical types we have in the cortex. It's about, I think it's about 300 altogether. But anyway,
How much?
300 or so mixed electrical morphological types. So you can see only here the diversity of the inhibitory neurons, which are much more diverse electrically. They are much more rich electrically. They are much more different than each other electrically than the excitatory cells. Excitatory cells are more boring. They tend to be like this more. Sometimes they're accommodating, but they don't have all this richness of repertoire of firing that the inhibitory neuron have. You don't understand this at all. Why do you take about 20%, it's not exactly 20% because you see that they distribute differently in percentage, yes, between the you see, but overall, let's say there is some people feel only 15% in the cortex, maybe even 11%, some 20%. Anyway, we are getting into the final numbers. This is, by the way, somatosensory cortex of young rats, age P14. Could be different in human and it could be different in giraffe. So this is a particular time and a species, yeah. But you can see there is this percentage, yes, proportion. So the question is, one question is really open and interesting question for research. Why do you put so much effort in making this small percentage of cells, let's say 20%, 15%, inhibitory cells, so rich electrically? And the rest of the 85%, excitatory neurons, are less rich in terms of the dynamics, yes, in terms of the capability to fire different type of spike patterns. I have ideas, but I have no idea why the system develops this way. This is, by the way, general rule, also in the hippocampus, that the richness of excitability, or the type of firing, is in the interneurons. I don't know why. Maybe it's energy reasons.
Different neurons?
Sorry?
Different neurons, or maybe even the same neuron in different locations?
No. So Yeah, so I didn't superimpose them. These are different neurons. So for example, you take this neuron, layer 4, basket tuft, inhibitory cell. You record from this, it will give you some kind of a typical firing pattern. Somebody else may have the same one, so I'm not saying it's one-to-one, as I said before. But this has a particular, this type of morphology cells has a particular firing type. This type of inhibitory cell will have a burst, it will be a burster. This will be a stutterer. This will be accommodating. This will be I don't know what, yes? All these 9 or actually 12 type of electrical, so we also did classification not only of morphology, but this is just a paper that came out from Charles Druckmann, who was part of the ICNC, a paper of classifying the electrical types objectively. So we again took all these firing patterns that I showed you before, which is of course very nice to put it like this, but this is not always what happens. There is a lot of noise within each type, so to speak. So the question, how many really objective types you have? You know that neurobiologists like to look at it, and they, by eye, of course, because they are classifiers themselves. We are classifiers, and we're saying, this is accommodating, not accommodating, burster, stutterer, we give names. But is this names objective? Or we just like people? So there was a very interesting meeting in Petia. Petia in Spain is where Monica Hall used to work at some point. So all the interneurons neurobiologists, those that work with interneurons, came to decide on a classification. It's called the Petia Convention. This was 2 years ago. And they fought about a week that this is different than this, or not different than this, and yes, different than this. My eye tells you that this is start later, and this is like here, and this is bursting here and not there. A lot of fight. So eventually we said, don't fight, let's do it objectively. Yes? There are new technologies. So we just did it. So I think it's finished now. At least from the database that we have. Again, this is limited. We have about 3,000 recordings from 3,000 cells. So maybe there is another cell that was not recorded because it's so rare. But from those that we have, we classified it, and there is some hierarchy here. If you want to look at the paper, you can.
Do we classify it based on degrees of each neuron, or can you also consider clusters of them?
So the classification in this case, what we did, we decided to, so I don't know if I answer your question, but at least I give you one thing, then I will, where is the light, then I really give you the rest of it. I always forget where is the light. So, yes, so what we did, in order to classify, we decided not to look, of course, on the pattern like this and say this pattern, this barcode, different than this barcode. We didn't do that. We said, okay, let's take this what Charles did. We said, let's say this is spiking pattern of one cell. Let's take what we call features. A feature can be a height of the spike, this will be one feature. The width of the spike, this will be another feature. The depth of the spike will be another feature. So the distance between the first and the second spike could be another feature. The IF curve could be another feature. So we had, as much as we could think, or shall we could think about 38 features that may characterize patterns like this. I just gave you 3 examples. And then he threw all the spikes that there were there. As I said, there were many spikes, from 2,000 to 3,000 cells. Many repetitions per cell, you know, in the experiments. Yes, many repetitions per cell, many cell types. But he didn't know anything. He threw everything into this basket of spikes, but he had these features, and he asked, you know, using PCA and some more sophisticated classification, whether just objectively you will get in this space of 38 features, you will get separated clusters, and he got them. And then you can go back and ask, okay, now I have this cluster, what cell it comes from, what morphological cell types it comes from. So that's why I can map again from this space of electrical clustering into the space of morphological clustering, and then so I can speak more wisely than fighting about whether this is different than this. Now we have that. But of course somebody can say, why did you choose these features? There may be other features. Anyways, but I don't know if I answered your question.
Yeah, I guess it's similar. My question was, how does
Oh, you mean when they work in ensemble, whether,
Yeah.
that's a deeper, okay, okay. So that's a good question in general, whether this particular feature that you see, so this is like taking the cell out of the environment, so to speak, separating it. And they do it, by the way, using this, because each cell is connected to others, and you are now sitting this is in vitro, no activity. And actually, they also use some drugs to disconnect the cells synaptically, so they don't talk to each other. So this is a property, or intrinsic property of the individual cell. It's the firing property of the axon, of the hotspot, of the nonlinearity in this one cell. This is your individuality. So you are a burster, you are a stutterer on your own. Now when I say stutterer or burster, I mean in response to a square current pulse. If I would inject sine wave, I don't know, I would get another behavior. So it's a very limited kind, but at least unified kind of what Henry Markov calls an E-code, electrical code. You inject different, you can do it also for different types of injections. You can inject a REM current, you can inject sine wave. So how do you classify? But in this case, it's classification based on step currents to each of these cells. So in the lab of Henry, if you go to a cell, you must go through a very fast characterization protocol. You have to inject this, you have to inject this, you have to inject this, and then you go to your experiment. So that's how we collected this. This is rare data on cortex, yes? And so that's what we took as our basis for classification. But the question, your question, if this is the question, is it relevant at all? So when I am now sitting in, I don't know, 10,000 cells talking to me, and I used to be Dan Segev, but now I want to impress you and you want to impress me, do I retain my individuality in this sense? So when I was a burster on my own, do I remain in some sense burster when the system is, or the system determines now that I'm not a burster, but I'm something else? That's a good question. If this is the question.
My question was more like, how do you know if you're measuring a small segment of the actual electrical type that's working as a whole, right? So
Give me suppose we are a little network here. Suppose we are a group of cells. You're a pyramidal cell, and he is, I don't know, an inhibitor, and I'm another inhibitor. And so, and you take me out, and you measure this, and you call me a name, you give me a name.
Right.
Okay, now what you ask?
Yeah.
What do you ask now?
Oh,
So this is what was done here.
right.
You took me out, you injected current to me, and I responded like this, and you called me burster.
So, I mean, so maybe then maybe what's really going on is that all of us are dancing. If we just look at what you're doing alone, it doesn't look like anything. It doesn't mean anything because it's really significant is the way the thing that's the way that it's gonna impact the collective, right? But it's not that you're gonna act differently necessarily. If we look at you within the group, you still look this. Well, no, I mean, you have to see it in that way, right?
Maybe. So are you asking really, suppose now, and that's of course an open question, so you have all these intrinsic differences. This is clear. Some cells have different channel types, and they express different ion channel types, and that's why this is firing like this, and the other one is firing like this. And we know even now the proportions. How many of these types there is in layer 2, how many of these types there is of layer 4, and all that. That's we know now. What we don't know, if this is your question, we don't know when the system is active as a whole. When you see a line like this in your visual cortex, or when the whisker system is now moved, how all these individualities relates or creates a specific dynamics of this particular sub-networks or a whole column in relationship to this.
Yeah, we could in principle do classification of electrical type for groups of 4 neurons, assuming that.
So I will take 4 neurons. Should I record from 4 neurons simultaneously?
Yeah.
And do what?
And consider that as
a unit.
Yeah.
As a unit.
Yeah.
But then, so instead of speaking about individual, you say the 4 now become a name, a unit.
Right.
But this, of course, there are many combinations of 4 now.
Right.
Should I take this one, and this other one, and How do I characterize them as a group when each one is different? That's what should I inject? What should I do to them in order to call them a name? What should I do? So what we did until now, we have to think what to do next. And I think we know what to do next. But what we did now is first to separate them and characterize them as an individual, just to call them by name or by class. Now we are trying to put them together into the whole circuit. Give the circuit an input, let's say thalamical input. I mean in the model now, I'm talking about the model, and try now to see whether there are emerging clusters of behaviors locally, and do they depend on these types or they are washed out because others are talking to it, because now you're getting input from 10,000 synapses. And so suddenly you start to behave. And there are some initially very interesting results in these simulations showing that some are very persistent in remaining themselves even when the network is doing crazy things, and some lose, so to speak, their identity and become very, very submissive to what is expected from them. But we are just starting to understand it. And again, you cannot, of course, use a mean field theory here and average everything, because I can show you that this is not always disappearing and there is no one unit come out of it. But what is remaining individually and what is disappearing in this context or another, we are just starting to explore it. Also experimentally, you can also do it experimentally, impale this cell individually and then activate the network and see if it remains like this or something else. But this is an interesting direction. I think really we need a little bit of water, no? And then come back, because I wanted to go into the computational aspect and I diverged myself. But so let's come in 5 minutes and then we'll continue. Yeah.
So I think what I wanted to ask was if we look at a computer, for instance, we might have so we can consider the computer as something like universal Turing machine, and there's only binary bits.
Okay.
And but when we give some input to a computer and then the computer responds in a high level coherent way, but there will be some number that's very relevant in our mind, that some number that we give to the program, and then this computer will respond, and there will be, at the binary level, this computer will also respond. But what might be happening is that 16 bits might be representing one information, and it's coherently responding to the program. But if you look at each individual unit, the frequency at which the bit flip might be different, just because
it's part of the system.
Yeah.
It's part of the. You're right. Yeah. So that's the metaphor I was trying to say, that when you are in a given context, you have to compute something. I mean, in the brain.
Right.
So this group of cells suddenly creates kind of a ensemble that represents something, and then your individuality behavior is now not anymore the relevant one, but rather than the group behavior.
Yeah.
But that's very, very difficult for me, of course, because you have to define the context.
Yeah, it's because if you look at 16 non-binary bits that represent the same information, there's nothing that's different within these bits, but when we actually look at the response of these bits to a program, you would see the difference in response, because the address that's assigned for each of these 16 bits to represent independent information is different. So they may not be necessarily different intrinsically, but just how they are assigned to represent an information.
But this depends on the context of the program. So this is not only a property of the cell, so to speak, but it is a property
of what is an independent unit.
But in the computer, as I see it, maybe I'm wrong, of course, in the brain, the independent unit could be a dynamical thing. For example, we can become, for this purpose, we can become a unit. And so now you and me and you, and so your individual bit is not that thing that is important, but something about our combined string, let's say, is important to do something. But in another context, you become a friend, so to speak, with him, and you share this. So it very much depends on what is the relevant context. That I look at you, in this context and try to... So here's the question is, again, we are not yet in the context, but people are doing this, we just spoke about this with the paper with Chaim now, the fact that the context itself changes, the fact that you suddenly stop and there's expectation and the visual system is not moving, yes moving, this would be the context, and so the individual cell would completely flip, inspiring or receptive field depending on what is the, in some sense is what you're saying, or not? Or is it related to that?
It's related, but I guess what I'm asking is, it seems like when we talk about neurons or brain, we clearly talk about very high level, like, when the network as a whole responds to some input, and then we also talk about the neural level, but it seems like a lot of times we don't consider the possibility that there might be another intermediate level that might be universal.
Okay, I see. The intermediate level is universal.
Right. It may not be, but it's a possibility.
But how will you, so you have to think, how would you, suppose there was such an intermediate state which behaves universally in some sense, how will you, what could be in principle? What kind of experiment or what kind of thing I should do to
figure out if there's such a thing?
In the computer, even in the computer, let's say that you are coming from I don't know where, and you look at your computer, there are only bits. And you claim that there is such a thing in the computer, let's say, and you ask me what should I look at? I don't know anything.
In order to...
I don't know in advance what was the program, so to speak. So but what should I, because an experimentalist I can, probe and do things. And as a theoretician I may build such a thing. But now that I have a system, what should I do to the system and look at it, either in silico or experimentally, what should I do in order to highlight there is this substructure? That's a difficult question.
So I tried to do something like this in Udall.
Oh, you did?
From a computer science friend of mine, I got a simulated computer which records full binary representation of a computer.
Okay.
You can give a program to a computer, and then you can look at how...
All the binary? Okay, fine, okay. Right.
Computation.
Okay.
It's really hard to figure out... I really
so that's the question about looking at the computer. That gives you all the binary...
Yeah, and we know from the way computer is built that the relevant unit that codes for independent information is 32.
Okay.
So if you look at the binary representation of a computer, it's very hard to figure out what's going on, but it's a lot easier to figure out what's going on if you
know that it's 32.
Yeah, because the dimension reduces significantly, and if you give a little bit of change to the program, what's changing, instead of looking at all these binary things that are flipping, you look at and there's one particular address that's changing all the time, and it's a lot easier to figure out, but figuring out what is the relevant unit is hard. Because even if you change the stimulus, quote unquote, or the number that you feed to the program, unless you explore the whole dynamic range of 32 bits.
Exactly.
Even if you know this is the address that this response or that it encodes the information together, there might be only like 3 bits that are flipping, and all the other... because if you're changing the number from 10,000 to 10,003, there's only...
I see, but they are not changing with the whole 32,
right.
but only some derivatives, but still the 32 is the relevant one.
Yes.
So the 29 plus the 3 are still...
Right.
Oh, no, now I see what you're saying. Not that I know what to say, what to tell you, how to do it experimentally, but I wonder if you see, we are getting some kind of a fundamental laws about the structure of connectivity. There are this very kind of embedded structures. There is a very strong connection between this layer to this layer, inside the layer less, and I wonder if I would tell you that this is imposing, if this is active, you must follow me because of this connectivity.
Yeah, I think it's
so structure, there are motifs very strong motifs embedded. It's not a random network. There are these motifs. Motifs can be from layer 1 to layer 2 and then again like this or some very strong motifs. And this might imply something about signal flow. Any way you do, this motif will take over. Let's say it's a hub. You must go through it. So, but we don't know yet. That's the thing.
Yeah.
That's the whole project of the connectomics, trying to find these motifs beyond statistics of one brain and another, and the complex structure, which may say something about the computational.
How's your student's project?
Sorry?
How's your student's project?
Which one of them?
Connectivity? On the connectivity of the network.
Oh, you mean a Eyal Gun?
Yeah.
Eyal Gun.
On the gun.
Also, it's very hard work because you see, Henry Markham is the only person in the world that records from 12 neurons together in a slide. We don't know anything about connectivity from in vivo. Impossible. So either it's recorded, you can do it from the connectomics reconstructed, but nobody did it as yet, and then you also don't know whether they are really connected. If there is a synapse, it doesn't mean that it's really a functional synapse. So Henry is the only one who does 12 patches. Again and again and again and again and again and again from layer 5. This is huge work to do 12 patches because you have to really patch 12 cells and stabilize them from the patch here. This goes on. It's very difficult. But so he's the only lab in the world that, so Eyal Gun has all these many 12 patches, only from layer 5. Only from layer 5. And he's trying now to statistically show whether there are, how frequent are these particular motifs beyond what is expected statistically. But statistics here you have to be very careful because you have 12 out of 2,000 cells and each one will take different 12. So the question, how many 12s you should take in order to be sure that what you find from this, I think he has about now 50 12s. 50 12s. How sure are you that the motifs you find are statistically significant? So we are doing a lot of learning statistics. This is very difficult, but there are some motifs that seems to emerge. One clear motif is if I'm connected to you, you are certainly by about 4 times than expected connected back to me. So it's not an independent multiplication.
But within the same layer of cortex,
nearby. So within 100 microns. If I'm connected to you,
the local connection.
Yeah. And also there is a very strong motif. If I'm connected to you and you are connected to her, there is a very good, very good, I mean about 7 times more than expected, and I would also. So we have a mutual friend. So it starts to create a certain social network. Yeah. He's using but it's really the beginning of trying to understand the embedded structure, and maybe this will give us some ideas about what is fundamental.
Was there a paper where Olaf Sporns, I think...
Olaf Sporns is doing it, Yeah, he's
science or something, I think science.
Yeah, but this is not local networks. This is, between regions in the brain. So Sporns is used...
probably about 10 years ago or something.
Sporns, are you sure?
Yeah. And there was actually I think he analyzed some data to look at the motifs, basically 2 or 3 neuron motifs. And he said that certain motifs occur very frequently, and this was a science paper or nature paper. And someone wrote a reply saying that if you just assume a small world network,
yes.
you get exactly the same thing. So it might just emerge from the fact that it's a small world network. And I don't know what happened.
I don't know if it's about local cortical structure, because Sporns, the book that he just wrote, it's a connectome one, but it's a connectome at the level of...
No, this was called, I think, Finding Motifs.
I think it's
I can look.
Maybe it's a song. Is it Sporns or is it...
No, Olaf Sporns.
Really?
Indiana.
Maybe I should look at this paper.
Yeah.
You can find it from...
Yeah, I'll
I'll be happy to see it. Because I know only these 2 papers about local circuits in the cortex by Song and Sasha Nelson, this is a Brand guy. And there is now this paper by Henry about these 12 patches. I think they did some statistical mistakes there, but we are now trying to do it better, more careful.
But
I don't know about this Sporns paper.
Yeah, I get you.
So yeah, please, it would be useful for me. So I cannot give you an answer about what we find yet. And shall we find it?
But when you say that you try to look at deviations from what's statistically true, what is the underlying model that, so are you assuming that the network is connected in the small world way? Or?
No, let's say that it's connected randomly for a second. Let's say that I have let's start. אנחנו מתחילים. ל-20 דקות. Okay, so we don't have much time, and that's why, not that it's not expected, what can one say in 2 hours about neurons? כי אנחנו ממש, אני ממש מתחיל. תמצאו עם האוכל, עם הכל, כי אין זמן, אין זמן. Okay, so I'll go very fast, just to show you what I wanted to tell you, not what I'm going to tell you, but what I plan to tell you. I just wanted to tell you now that just completing this issue of classification of single neuron types, there is a very nice project, both started with Henry Markham in Weizmann, but now Sasha Nelson in Brandeis are trying to really do the ultimate classification using genetic profiles or expression of genes in different cell types, trying to to see how many cell types in terms of genetics, because this of course reflects on the electrical types, which is also is a signature of which genes or ion channel genes.
Proteins, not genes.
Yeah, protein. Sorry, this is this is the protein expression, but then they are also going to the genome level, see which gene is being activated or expressed, and what protein is being expressed. So they are doing this genome, and so this is the work of Sasha Nelson especially. I don't understand how they do it exactly, I must say. There is a single cell PCR, and there are new technologies that I don't understand, but there is this ultimate attempt to ask how many genetically type of cells you have in the cortex, whether it will be 200, 300, or I don't know what, and what will you do with all this information, that's another question. But anyway, I just wanted to highlight that, and I want to mention very fast some ideas about computing with dendrites, because that was really what I wanted to aim at when I spoke about biophysics, differences in electrotronic distance, and so on. So I cannot go all over that now because of the limitation of time, and even if I had time, I wouldn't be able to go. So I just want to mention some old ideas, and also eventually point to this very new Nature paper by Jackie Schiller from Technion, who did, I think, a beautiful, really, really beautiful work, something we very much expected and wanted somebody to do, an mathematician. And she did it, this is the first round of ways to do it, and in this case what she did is the following. She took a cell, a pyramidal cell, from the whisker system of the mouse. This cell, she characterized the receptive field, or the sensitivity of this particular cell type to, in this case, orientation of movement of the whisker. So it was an orientation selective cell to the direction of whisker movement. And then she wanted to ask, what do dendritic nonlinearities do, or how do they contribute to this computation? So you characterize a particular computation, I am a cell sensitive to, I don't know, this direction. And then what she did, and this is due to new kind of drugs, apparently there is a drug that you can inject intracellularly only to this cell. So only this cell now is affected by this intracellular drug, and this particular drug, which is P something 816, I don't remember, blocks NMDA synapses, NMDA nonlinearity. Only, specifically, from the inside. And so the input in terms of synapses, of course, changes, and some subset of synapses are now being affected, not only synapses, but I didn't speak about, there is this, what is called the dendritic NMDA spike. So she succeeded to block the NMDA spikes from within this cell. And she shows how when she eliminate these particular nonlinearities, there is a complete shift in sensitivity to direction. So this is a claim that the cell itself, the nonlinearities of the cell itself, play a key role in eventually shaping the computation that this cell is doing. But again, I don't have time, but this is, I think, the most ultimate work that we have today about showing what are the local properties of cells have to do with the eventual phenotype of computation that the cell is doing. But I don't want to say much about this, just to mention the history. because I don't think we should speak about today without some historical ideas. This is, the dramatic McCulloch and Pitts. This is the McCulloch, and the McCulloch and Pitts was actually the first one to think that the neuron is doing some kind of computation. And this inspired computer science. 43. This is the classical paper of McCulloch and Pitts. Really classical. The classical paper of McCulloch and Pitts is from 43. Again, working very beautiful, very influential paper on computer science, mostly, not on neuroscience. Neuroscience didn't like this at all. This is the McCulloch and Pitts neuron, the M and P neuron. So this is the M and P neuron who can perform certain logical operations. So if this is excitation and this is inhibition, they claim that you can do some kind of an and not or operations, logical operation or multiplicative operations using this kind of McCulloch and Pitts neuron, because if this and this operates and not inhibition, then you get an output. So this is McCulloch and Pitts. But they showed that you can build a universal machine using this kind of operation. So these 43 papers are really fantastic in MIT, and this is really ignite computer science. So neurons ignite neuroscience. The idea that neurons are off on 0, 1, and they do this kind of and not and excitation, not inhibition. So you should know that the history of science, computers were very much affected by some ideas that came from neurons, but neuroscience was not affected by this idea.
Not yet.
What?
Not yet.
Not yet. Maybe yes. But anyway, those that were affected by this, just to give you the history, is Koch and Poggio, who were in MIT at that time. So this is 82. And then they said, okay, this is McCulloch-Pitts point neuron, but what would have happened if you have a distributed neuron? So we know that it's distributed, and then of course the geometry or relationship between inhibition and excitation plays a key role in the type of logical operation you can do. Because this inhibition, in principle, can veto all the other excitation that is more remote from it. So it's E3 and E2 and E1 and not I1 and so on. So the geometry of inhibition is important here. So this is this kind of operation. So that, again, I don't want to go into this form because that's now very classical work of trying to extend McCulloch and Pitts into a distributed system, showing that the location of inhibition relative to excitation, especially in branched dendritic tree, becomes very critical because, for example, this inhibition can veto everything that is on the path between this and the soma. If there is an inhibition on the path, it can veto all this, but it will not veto everything that goes here. So the location, and if I had an inhibition on the axon, then it will be a global veto because I don't care where it comes from. I kill it on the spot where I want to generate an output, yes or no. So anyway, this is Koch and Poggio logical operation ideas of single neurons due to geometry and location of things.
Poggio is a good friend of Shimon
Of course.
Ullman.
Both of them were students of David Marr. So David Marr was the father of everybody in MIT in terms of neuroscience. He died very early on. He wrote this ultimate, very beautiful book called Vision. And so Poggio, Shimon Ullman, and then Christoph Koch, who was a student of Poggio, all came to MIT following David Marr, who was also a very good cellist, a genius, complete genius. He died from cancer very young, and if you read the introduction of his book, you see that he thanks his doctors for enabling him to finish the book. Very touching, very special person, David Marr, British. Okay, so this is all ideas that I don't want to go, but I think you should, when you speak about single neuron computation, you should know that it came from there. And there is also a lot of work, as you heard, I don't know if you heard, or maybe you didn't, recently by Michal, who is a candidate for the ICMC. She's working on directional selectivity of retina ganglion cells. You know that in the retina of certain species, some cells, some output cells, this is the third level ganglion cells, are directional selective, and so they prefer, so if you give a bar moving or a grating moving in this direction or another, Sometimes there will, in some direction there will fire spikes, in other direction there will not fire spikes. So the direction selectivity, the computation of direction selectivity is already done at the level of retina. This is a local network. This is independent of the top-down effect because there is no much top-down effect onto the retina. So this is an isolated retina. So something about these 3 layers local circuit already do some computation, making cells sensitive to this direction of motion, this direction of motion. So you see this direction of motion. Something about your retina is already telling the brain that that's what happens. And so the idea was, and this, because of this, this is an old work, this directional selectivity of ganglion cells in the retina, induced Will Roll, who developed the cable theory, to try to suggest a way to do this kind of directional selectivity based on dendrites and synapses. So this is from '64 paper, where he said the following, and this is now connecting biophysics of synapses and dendrites to computation. So this was his idea. So this is the cell body, this is my cable, this is my dendrites. And synapses, excitatory synapses can be anywhere. So these are the excitatory synapses, the black are the excitatory synapses. And so he claimed that, not only claimed, he showed by simulations that the direction, so the temporal sequence of activation of your excitatory synapses is reflected differently at the cell body depending on how, in terms of temporal sequence, you activate the synapses. For example, suppose you start with do excitatory, so you do tak, 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 tak. And then you use the same synapses, absolutely the same synapses, but in reverse order in time. Tak, 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 tak. So distal to proximal or proximal to distal. But it's the same set of synapses, just different order of timing, of spatio-temporal timing. Yes? And then when you recorded the cell body, you can see from what you already know from the biophysical part of my talk, that if you recorded the cell body and you do A, B, C, D, sequence of activation, you will see that the nearby synapses are seen at the cell body very fast, no delay, small delay, we spoke about delay, okay? And then on top of this shoulder, so if it would have been only this, there would be a low somatic EPSP and decay. But because in time there is this activation which appears later at the cell body, you start to climb, or sum, on top of the shoulder of this EPSP. So second EPSP, third EPSP, fourth EPSP, as it's seen at the cell body, is summing on the shoulder. If you do the opposite direction, there is a delay because you start distally. There is a delay at the cell body, but if the velocity, if the delays are correct, then this is summed on this, is summed on this, is summed on this, and you may build up larger voltage. Just because of the location and the delay, that's all. Nothing magical here, yes? If it would have been current sources, then the integral below these 2 composite EPSP, not one single EPSP, composite EPSP, will be the same integral because the same charge will arrive to the soma, but the temporal structure of it will be different.